Hello students, myself Dr. Mohammad Zafar Iqbal. In this video we are going to continue with the genetic engineering, that is the recombinant DNA technology, and in this video we are going to discuss about the gene cloning. In the earlier videos we already discussed about the how to extract the DNA and how to analyze the quality and quantity of the DNA molecule, and in the second video we already discussed about the different types of cloning vectors. We have especially given the emphasis to the plasmid based vectors because most of the time in the majority of the recombinant DNA technology plasmid based vectors are used. That's why we have concentrated on the plasmid based vectors, and today we are going to continue with the same thing, and we are going to see how the gene cloning is actually done. Before we get into this particular topic, let us set some of the important learning objectives. And the first learning objective for this in this video is the to understand the meaning of the nucleases. We will try to understand what exactly the nuclease means. This is very, very important term, and I think you might have heard about different types of the viruses which come under the different type of the enzymes which come under the category of the nucleases. And we are going to study a particular specific type of nuclease called as the restriction endonuclease, because this particular enzyme is very, very important in the recombinant DNA technology, and that's why we are only interested in this particular enzyme. Just for brief introduction, that's why we are going to discuss something about the nucleases. Then we are going to discuss about the preparation of the competent cell. Majority of the time we will find that whatever this particular bacterial cells are there, they are very, very poor transformers. Actually, they don't receive the foreign DNA molecule. That's why here we are going to use some of the techniques, scientific methods, and by using the scientific methods we are going to increase the efficiency of the transformation. That we are going to study in this particular video. Then we are also going to discuss about the general scheme of cloning, how exactly the cloning is done. That we are going to see in some of the slides, and at the end of this particular video we are going to see the screening. If you take a number of cells, some of the cells may be recombinant and some of the cells may not be recombinant. Then how to identify which cells are recombinant and which cells are not recombinant? For that we have got very, very elaborated techniques are there, and in this video we are going to discuss about only two screening techniques to identify the recombinant cells. Okay, before we get into this particular topic, let us discuss about the nucleases. I think most of the students might be knowing the nucleases are nothing but these are the enzymes which are specifically meant for the degradation or digestion of the DNA or RNA. If you have an enzyme, if it has got an ability to digest the DNA molecule, then we call it as it is a type of the nuclease. And if there is an enzyme which has got an ability to digest the RNA, digest means cut the RNA molecule, then it is called the nucleases. There are certain nucleases which are specifically meant for the degradation of the DNA, and there are certain enzymes which are specifically meant for the degradation of the RNA. And if you take both the DNA as well as RNA, there are two categories of the nucleases. One is called the exonuclease, second one is called the endonuclease. As the name itself suggests, if you take exo, what is the meaning of exo? Exo means outside, and what is the meaning of endo? Endo means inside. It means that whatever these nucleases are there, these nucleases have the ability to break the bond, break the phosphodiester bond from the ends. Take for example, this is the diagrammatic representation of the exonuclease. Here you can find that these are the ends of the DNA molecule, and this is the other end of the DNA molecule. And if you take the exonuclease, you can find that they have the ability to digest the DNA molecule from the end. First, this particular base will be removed. After that, this particular base is going to be removed, and after that, this particular base is going to be removed. And whatever the exonucleases are there, there are two types of the exonuclease. One is called the 5' 3' exonuclease. It means that if they have the ability to remove the nucleotides from the fifth end, fifth prime to third prime, then they are called as the 5' 3' exonucleases. And there are certain enzymes which have the ability to remove the nucleotide from the other end, that is from 3' to 5', and such type of the exonucleases are called the 3', 5' exonucleases. In this way, this is the way these are the exonucleases. And today we are not at all interested in the exonucleases, we are interested in the endonucleases. And what are endonucleases? If you just compare these two diagrams, you will come to know that here the enzyme is actually breaking the phosphodiester bond from the end. But in case of this particular endonuclease, you will find that the endonucleases have the ability to break the phosphoroester bond which are in internal position. Take for example, this is the end of the DNA molecule and this is the other end of the DNA molecule. And if you take the endonucleases do not remove the nucleotides from the end. Instead, they break the phosphodiester bond into in the internal position. Actually, that's why they convert this DNA into two fragments. Now you can see that this will become, after the addition of this particular phosphodiester bond, then you can find that it will become one fragment and it will become the second fragment. And in this way, you can find that whatever this particular endonucleases are there, the endonucleases are nothing but they are the enzymes which break the phosphodiester bond in the internal position. In this way, you can find that there are two types of the exonucleases. One is the exonuclease and second one is the endonuclease. In this video, we are not at all interested in case of all the types of, if you take the endonuclease, so many different types of the endonucleases are there. But in this video, we are only interested in a very specific type of the endonuclease, what we call it as a restriction endonucleases. What are restriction endonucleases? The restriction endonucleases are not a simple type of the enzyme. They are very special type of the enzymes. And in what way they are special? They have got three very important features. The first feature is they have the ability to identify a recognition sequence. That is, they have, if you take a complete DNA molecule, within that particular DNA molecule, you can find that they have the ability to identify a particular area or a particular given segment as the recognition sequence. And once the recognition sequence is identified, then it has got an ability to break the phosphodiester bond exactly at a particular bond is going to be broken. And that's why that particular bond or that particular area is called as the restriction site. And whatever the restriction endonucleases are there, the restriction endonucleases, they exhibit the specificity. Since they have these three properties, that's why we call them as a restriction endonucleases. If you take the other enzymes, you take the example of DNase 1 or DNase 2, what they do? They also have the ability to break the phosphodiester bond in the internal position. The only thing is that the non-specific. Non-specific means in one particular experiment, they may break one internal bond. In the second, if you repeat the experiment, then you will find that they may break some other bond in the internal position only. But as far as the restriction endonucleases are concerned, you can find that if you use the restriction endonuclease, all the time they are going to break only one bond. That is the point we call it as a recognition site, and that is the restriction site. And where exactly that restriction site is present, that restriction site is present in the recognition sequence. In this way, you can find that these are the three very important type of the unique features which are present. And if these three unique features are present, then only we call that particular endonuclease as the restriction endonuclease. And only this type of the enzymes are useful in case of the what we call it as a recombinant DNA technology. Then if you move to the next this thing, now if you take the restriction endonucleases, till today, as many as more than 5,000 different types of the restriction endonucleases have been identified. And once you identify so many number of restriction endonucleases, ultimately all of them do not have the same features. They may differ from each other in certain features, certain characteristic feature. Based upon that particular characteristic feature, we have these restriction endonucleases have been classified into four main types. That is called as the type 1 restriction endonucleases. Second one is called as the type 2 restriction endonucleases. Third one is called as the type 3 restriction endonuclease, and fourth one is called as the type 4 restriction endonuclease. And in this particular recombinant DNA technology, all of them have got all those features. Only thing is that some kind of differences will be there. And out of all these four different types of the restriction endonucleases, the type 2 restriction endonucleases are very commonly used in case of the recombinant DNA technology. You take the type 2 restriction endonucleases, they are the enzymes mainly used in case of the recombinant DNA technology. And if you take total number of restriction endonucleases, type 2 restriction endonucleases so far identified as there are as many as more than 3,500 different types of the type 2 restriction endonucleases have been identified so far. And what are the features of these restriction endonucleases? They have the ability to carry out the restriction reaction. They also have the ability to that is the modification. Modification means transferring the methyl group. That is called the modification. And whatever the type 2 restriction endonucleases are there, these enzymes are completely dependent upon the presence of the magnesium divalent. If the magnesium divalent is present, then only these particular enzymes will be able to perform their function. And magnesium divalent is not present, then they may not be able to perform their function. And if you take the restriction endonuclease, if you take a complete holoprotein molecule, you will find that there are three subunits are there. One is called the R subunit, and this is the restriction subunit. Restriction subunit. Then you will find that this is a M subunit, and this is the modification subunit, and this is called as the S subunit, and whatever the subunit is, this is called the specificity subunit. And since if you take all these three different types of units, you can find that each of them have got a particular function. The specificity will identify the recognition sequence, and once the recognition sequence is identified, then the R subunit, that is the restriction site, will be cleared. Then you can find that whenever the modification is required, that is the addition of the methyl group, then the M subunit will be used. And in this we can find that there are all the three subunits are present, and each subunit has got a particular function. And whatever this particular M subunit is concerned, we already know what is R subunit. R subunit means this is the restriction subunit which is responsible for cleaving the phosphodiester bond. And whatever the S subunit is there, the S subunit is mainly responsible for the identifying the recognition sequence. And now, if you take the M subunit, you can find that the M subunit is mainly responsible for the methylation of that is the methylation reaction, and that is the addition of that is the transfer of methyl group from adomate. And where that particular, who is the donor of the methyl group? The methyl group will always be donated by means of a adomate, and that particular methyl will be transferred to, that is methylation occurs at cytosine at 5th position, cytosine at 4th position, and cytosine, that is the adenine at the 6th position, cytosine at 5th position, cytosine at 4th position, and adenine at the 6th position. These are the three different areas where exactly you will find at the modification. Modification means that is the addition of the methyl group will take place. And whatever this particular restriction endonucleases are there, the restriction endonuclease will always identify the palindromic sequences. Palindromic sequences are nothing but they are the symmetric sequences. And what are the palindromic sequences? Palindromic sequence is nothing but if you read in one direction or if you read in the opposite direction, it will be same. And that's why it is called the palindromic sequences. And if you take this particular the example of a deed, take for example deed, I am writing here D-E-E-D. And if you take this particular word, you will find that if you read it in this direction, or if you read it in the opposite direction, it is same. This type of the words are called as the palindromic sequence. Another example is the Gadag. If you take the example of a Gadag, this is the place in Karnataka, and if you take this word, here you can find that if you read this in this direction or in the opposite direction, it is same. If you read here G-A-D-A-G, and if you read here G-A-D-A-G, this is also called as the palindromic sequence. And whatever the restriction endonucleases are there, type 2 restriction endonucleases are there, the type 2 restriction endonucleases will always identify the palindromic sequences, and that's why they are very, very specific as compared to the rest of the. Why we use the type 2 restriction endonuclease? The answer is that these are the enzymes which have the ability to identify the palindromic sequences, and that's why it is much more efficient as compared to the rest of this thing. And within the restriction sequence, there is always a restriction site will be there, and that's that whatever this particular restriction site is there, the restriction site will either be within that particular recognition sequence or slightly near to that particular recognition sequence, but at a fixed position. That is also very, very important. If you take this particular point, the restriction recognition sequence, the restriction site will always be present either in the within the recognition sequence or slightly away from the recognition sequence. It means that at a fixed position. If you repeat this particular experiment to take the restriction endonuclease, type 2 restriction endonuclease, and digest the DNA molecule for n number of times, in all the occasions you are going to get the same type of the fragments will be generated, same fragment, same number of fragments, and if you take the length of each fragment, the length of the fragment will also be identical. That's why it is more reproducible. That's why you can find that this particular type of the enzymes are much more convenient in case of the that is the recombinant DNA technology. And if you take the type 2 restriction endonucleases, there are as many as 10 subclasses. Again, I said that if you take total 5,000 restriction endonucleases are there, and out of 5,000 restriction endonucleases, somewhere about 3,500 restriction endonucleases they belong to type 2. Then ultimately you will find that they may differ from each other, and that's why when you minutely analyze the features of this particular restriction endonuclease, type 2 restriction endonuclease, further we can classify them into 10 subclasses or 10 subgroups actually. And some of the very common examples of the type 2 restriction endonuclease are nothing but is the EcoR1. If you take the EcoR1, this is the restriction endonuclease which is very, very commonly used throughout the world. You go to anywhere, if some recombinant DNA technology cloning experiments are going on, in that laboratory they must be using the EcoR1. The second very common type of the enzyme is the HIND3, then HEA3, BCG3. All these are nothing but they are the very common type of the type 2 restriction endonuclease, and all of them are the come under the type of the type 2P restriction. Type 2P means palindromic restriction the enzymes. Now we are going to identify what exactly the recognition sequence means. In the earlier slide, we have used the word called as the recognition sequence. We have used two words. One word is called as the recognition sequence, and second one is called the restriction site. What is the recognition sequence? I have said that the restriction endonuclease will identify a particular region of the DNA molecule, and that particular region of the DNA molecule is called as the recognition sequence. For ECO1 GAATTC, this is the recognition sequence. If this particular sequence is present, then only that will be identified for as a substrate for the cleavage, for the cleaving. And if this particular DNA, take for example a DNA, if it doesn't have this particular sequence, then that particular enzyme will never break that particular DNA molecule. It means that in order to break the phosphodiester bond, the EcoR1 should have this particular recognition sequence, and within this particular recognition sequence is called the that is called the recognition sequence, and this is a palindromic sequence. How can we say palindromic sequence? You read from this direction or read in the opposite direction. This is GAATTC, and if you read in the opposite direction, the second standard GAATTC. Remember that in case of the DNA, whenever we say DNA, it is always we mean by a double stranded DNA molecule. That's why this is the one strand of the DNA molecule and this is the second strand of the DNA molecule. If you read in one direction, it is GAATTC, and in the opposite direction, it is GAATTC. That's why this is nothing but this is called as a palindromic sequence. And now you can see this particular arrow mark. Can you see this particular arrow mark? This is the one arrow mark and this is the second arrow mark. This is called as the restriction site. I already said that whenever you use the recognition, the type 2 restriction endonucleases, they always break at one particular area, at one particular bond. They are going to cleave a specific type of the phosphodiester bond. And whenever you take all the EcoR1, you will always find that within this particular recognition sequence, they break the phosphodiester bond which is present between G and A. If you take between this G and A, there is a phosphodiester bond is present, and whatever the EcoR1 is there, the EcoR1 will always break this particular bond. And in the same way, if you in the other strand, this particular area, that is the immediately after this particular between the G and A, the phosphodiester bond is present. This particular enzyme is going to break this particular phosphodiester bond. In the similarly, you take the example of a BAMH1. This is the recognition sequence for the BAMH1. If you just observe the recognition sequence of the EcoR1 and see the recognition sequence in case of the BAMH1, they are different from each other. This is the substrate or the recognition sequence for the BAMH1. And if you take this particular recognition sequence, this is the recognition sequence for the EcoR1. And if this particular sequence is present in the DNA, and if you use EcoR1, nothing will happen. That bond is not, that particular DNA is not going to be cleaved. And in the same way, if you use the BAMH1 and if this is the recognition sequence, then automatically you can find that this particular enzyme is not able to break the phosphodiester bond in the GAATTC. That's when you find that this particular enzyme is specifically meant for this recognition sequence, and this particular enzyme is specifically meant for this recognition sequence. And in the same way, this is the HAE3. Now, if you take the HAE3, you can find that this is the GECC is the recurrent sequence. Again, it is also palindromic sequence. You take this particular sequence, this is also palindromic sequence. Whatever three examples I have taken, all these three examples are nothing but they are the examples of the palindromic sequences. Now, this is the common. Now, if you carefully observe these two enzymes and this particular enzyme, you will notice one particular important difference. Here you can find that the cuts which are going to be generated will produce a different type of the DNA fragments, and if you take in these two examples, and in case of the HAE3, you will find that whenever you cut is generated here, you can find that different type of the fragment is generated. Based upon this, we have two different types of the fragments are generated. One is called as the sticky end fragments are generated, and second is called the blunt ended fragments are generated. And whatever the sticky ends are there, they are also called as the staggered ends. Why we call them as a staggered end? Because if you take this particular now EcoR1, this is in between G and A, that was the restriction site. And once the bond is broken, now you can find that this will become a separate DNA. This will become the separate DNA fragment actually. Now, if they come close to each other, then they will stick together actually. And because of this particular property, we call them as a sticky ends. They have the ability to make the hydrogen bonds between. If both of them come together, then A will make a hydrogen bond with the T. This A will make a hydrogen bond with this T. and this T will make a hydrogen bond with A, and this T will make a hydrogen bond with A, and in this you can find that it will again completely seal with each other. In the same way you can find that if you take the example of a second example, that is the BAM H1, here also you can find that this is also another example for the sticky ends, and whatever these particular two examples are there, both of them are nothing but they are the examples for the sticky ends. Here you can find that this is the staggered ends are created in this particular fragment. Here also you can find that the staggered ends are created here. And if you take the second type of the fragments which are generated after the digestion of the DNA, that is the with the DNA restriction endonuclease, is that blunt ended fragments are present. And we have already taken the example of a HAE3. Here you can find that the bond is present between GG CC. If you take GG and CC is present, and the bond is going to be phosphodiester bond is broken in the after G and C. Here also same bond is going to be broken. That's why you can find that if you observe this particular fragment, you can see that it doesn't have a overhangings. Single stranded overhangings are not present. Here you can find that some single stranded overhangings are present. And but in case of this particular HA3, you'll find that no such type of the overhangings are present. And whatever these particular blunt ends are there, these ends are called as the blunt end, and that's why it is called as the, it is also called as the flush ended DNA molecule. In this way you can find that this type of the fragments are generated. In this way you can find that whenever you digest with the that is the distinct endonuclease, you can either, depending upon the type of the enzyme, either you may get a sticky end DNA fragments or you get a blunt ended DNA fragment. And out of these two, which one is better for the recombinant DNA technology? That is the cloning experiment. And between these two, you can find out the sticky ends are always useful, because if you take the blunt-ended DNA molecule, making a ligation reaction is very, very difficult. Whereas in case of the sticky ends, you can find out the ligation reaction is very, very easy. That's why the sticky ends are much more convenient as compared to the blunt-ended DNA, that is the enzymes. That's why in the most of the occasions you can find out the scientific community who are doing the cloning experiment, they use this type of the fragments, this type of the DNA restriction endonuclease, which generate the sticky ends or the staggered ends. And rarely blunt-ended restriction endonuclease which create blunt-ended ends are actually used. This is one point. And how, if you have a blunt-ended DNA molecule, and if you want to continue the cloning experiment, what is the that is the, how can we overcome this particular problem? We can overcome this particular problem by using the linker DNA molecule. Several different types of the techniques are there. One is called the, we have to attach this particular DNA fragment with the linkers, or we have to attach this particular DNA fragment with the adapter, or we have to link this particular DNA fragment with the homopolymeric tailings actually. In this way, we can find that we can convert the blunt-ended DNA molecule into the sticky end. That's why once the sticky ends are formed, then you can find that it becomes very, very easy for the ligation reaction. That's why most of the time we can find that we use the enzymes which produces the sticky end. And if you are forced to use the enzyme which produces the blunt-ended molecule, then you have to go for a additional techniques. And by using that particular additional techniques, we are going to convert the blunt-ended DNA fragment into the sticky ends. Okay, now the next step is the one more important point is that whatever the enzyme which are using for digestion of this particular genomic DNA, same enzyme should be used for the digestion of the vectors actually. That I have already told in the one of the videos. And now we are going to move to the next step, that is the preparation of the competent cells. How to prepare the competent cell? I already said that in case of the bacteria, they do not the recombination is only possible by means of a three method. One is called the transformation, second is called the transduction, and third is called the that is the conjugation. These are the three ways by which the bacteria undergo recombination. And out of these recombination, one of the technique is very much useful in case of the recombinant DNA technology, or in case of the cloning, that is called the transformation. But if you take majority of the all bacteria do not exhibit the phenomena of the transformation, because bacteria got three different ways by which they can undergo recombination. They can undergo recombination by means of the transformation, they can undergo recombination by means of a conjugation, or they can undergo recombination by means of a transduction. That's why there are three different mechanisms are present. That's why they may undergo transformation, they may not undergo transformation. And now in the laboratories, one of the very, very commonly used type of the bacteria is the E. coli. We use the E. coli because we know that particular genome very well, and we can manipulate it in any way. And because of that, we most of the time what we do is that we use the E. coli. But if you take the E. coli, the E. coli are very, very poor transformants. They undergo transformation very, very poorly. They are not inefficient. The transformation is very much inefficient in case of the E. coli. That's why we have to convert that particular inefficient transformation to efficient transformation. That particular process is called as the preparation of the competent cell. How we are going to help in the process of the transformation, that we are going to do. And in order to do that particular, now what we have to do, we have to insert the vector along with the recombinant DNA into this particular bacteria. And that's why we are using this particular thing. We have got two different methods are there. One is called the chemical method, and second is called the electroporation method. And if you take the chemical methods are generally used because electroporation techniques are very, very harsh. And sometimes even if you take the machine, the unit which comes, that is very, very costly. Most of the time we can find that very, very high voltages are used for a very, very short time. And during that time we can find that the plasma membrane will expand because of the shock, heat shock, that is the current shock. And once it is expanded, then you can find that DNA molecule will enter. In this way, you can find that the transformation is carried out by means of the electroporation technique. But I have already said that this particular technique is not very, very common, because one is the technique is very, very expensive, and second one is that most of the time you can find that it leads to the death of the bacteria. Actually, that's why you can find that most of the scientists they not prefer this particular technique. And one of the very, very common and which can be done in any laboratory is the chemical method, that is by using the calcium chloride. We use a particular chemical called as the calcium chloride. What the calcium chloride will do? The calcium chloride, once you treat a bacteria with the calcium chloride, then what will happen? The calcium chloride will make this particular DNA molecule, that is the plasmid DNA molecule, to go and adsorb to the surface of the bacteria. Only that is going to be done by means of the calcium chloride. It is not going to open the that is the pores of the plasma membrane. And once you immediately transfer that particular calcium chloride treated bacteria at 42 degrees centigrade, automatically you can find that you are changing the heat, you are actually changing the temperature. And because of this particular sudden fluctuation in temperature, automatically I can find that the bacterial cell will swell. And during that time we can find that it will open this particular, you can see that here in this particular bacteria. Now if you see this particular plasma membrane, you can see that complete layer is present. And now if you see this particular layer, you can find that because of the expansion there is a pores are present, openings are there. And by using this particular pore, you can find that the plasmid DNA along with the recombinant DNA will enter into this particular bacteria. This is called as the preparation of the competent cell. How we are going to prepare the competent cell? By in this way, in this procedure, we can prepare the competent cell. That is the one of the very, very important thing. Now we will go to the general scheme of cloning, because since this particular video is meant for the to discuss about the cloning at the end, why we have done the we have studied about the cloning vectors, we wanted to go for the cloning experiment. Why we have studied about the restriction endonucleases? Because we wanted to use those enzymes for the cloning experiment. Why we have isolated the DNA molecule? Because we wanted to use that particular DNA for the cloning experiment. Now here in this particular diagram, we are going to see the briefly about the how exactly the cloning is done. It is a very, very elaborate technique. Already in pieces we have discussed here and there, but just in order to compile that particular information, we have to take for example, this is the human cell, and we want to clone human gene into the bacteria. What we want to do, the aim of this particular experiment is to clone. We want to take some gene from the human being. Maybe in this particular example, they have given the gene which is responsible for the production of the growth hormone. Okay, now you have taken the gene of the growth hormone. And how you have taken this? You have taken the genomic DNA, you extracted the genomic DNA by using EcoR1. You have digested that particular DNA, because your same enzyme is going to be used for the to cut this particular plasmid also. Then you can find that once it is done, then you can find out what you are going to do. Now you are going to insert this particular human growth hormone gene into this particular plasmid. That is the now you have done this. And after that, what will happen? Now what will happen? The next important thing is that we are going to introduce this particular now this particular DNA is called as the plasmid is called as the recombinant DNA. Why? Because it has got along its own genome. Here you can find that this is the actual genome of the plasmid, and now after the digestion it has opened. Now you have your piece, that is the gene of your interest, has been inserted into this particular vector. Now what you have done, now once it has again became a circular DNA molecule, now it has got a extra DNA molecule. What is this extra DNA molecule? This extra DNA molecule is nothing but this is the gene for the growth hormone, human growth hormone. Okay, now what you want to do? You want to introduce this particular plasmid into the bacteria by using the what we call it as a by means of the competent cell technique. Now you can find out it is inserted. Now you can find out whatever that particular gene of interest, it has been incorporated into the plasmid, and whatever that recombinant plasmid is there, that recombinant plasmid has been introduced into the bacteria. This is the way by which we are going to introduce the foreign genes into the bacteria, into some other organism. That is called the cloning. And after that, what we are supposed to do? We are supposed to screen, because once you complete this particular thing, there are three possibilities. One of the possibilities is that it can have a recombinant. Second possibility is that it may not have a recombinant. Third possibility is that whatever the recombination there, the recombination might have taken at some other position instead of at that is the targeted site. You wanted to clone that particular gene into this particular. You wanted to inactivate some selectable markers. But what happened is that it has been incorporated to some other region actually. That's why in every cloning experiment, there are three possibilities are there. The first possibility is getting the recombinant DNA molecule. The second possibility is that there is a possibility of the non-recombinant. Without any DNA, foreign DNA, it can become circular. That is the second possibility. And the third possibility is that the recombination will take place, but that recombination will take place at a place which cannot be identified by means of a screening technique. That's why you can find out this is the three different possibilities. Now what we are going to do, the first method, very simple method, is the, since where exactly we have inserted our gene of interest, we have inserted our gene of interest into the selectable marker. This is the selectable marker of the plasmid, and once the gene of interest has been incorporated here, then what will happen? It will not be able to produce its gene product actually. Here it is able to produce the gene product because the gene is intact. Okay, take for example, if this is the gene for the ampicillin, and if it is completely intact, then it will produce the ampicillin resistance. So it will give the property of the ampicillin resistance. And in case of this particular example, you can find out whatever our gene of interest, that is the human growth hormone, has been inserted into selectable marker, and once it has been done, automatically can find that this gene has been converted into two fragments. One particular fragment of this gene is present here, the second gene is present, part is present here, and that's why it will not be able to produce the selectable marker. That's why you can find out it will not give any this thing. That's why you can find out just by means of this particular simple technique. If you get that is the gene product, then it is a non-recombinant. If you are not going to get any function of the selectable marker, then it is a recombinant DNA vector. Then you can find out the second type of the screening is called as by using the LAG-Z. Sometimes you can find out if you use the vector called as the PUC18, PUC19. What we do, we don't use the ampicillin or a tetracycline as a selectable gene. We use LAG-Z dash, and whatever this particular LAG-Z dash, if you take this particular LAG-Z dash, this is the part of the gene, and if this particular part of the gene, if you want to have this particular part of the gene, you must have this thing is the activator should be there, and that's why you can find out whatever this particular experiment is there. If you take this particular experiment, you can find out whatever it is done by means of a XGAL plus IPTG. This is called the XGAL and IPTG. What is XGAL? XGAL is nothing but it is 5-bromo-4-chloro-3-indolyl beta-D galactopyronoside. And whatever this particular product gas XGAL is there, it is nothing but it is an analog of the lactose. Instead of using the lactose, what we are going to do? We are using the XGAL. And what is XGAL? XGAL is the 5-bromo-4-chloro-3-indolyl beta-D galactopyronoside, and that will work as a analog of the lactose. And second thing, what we are going to use? We are going to use a stimulator or the activator of the enzyme, and that is called the IPTG. What is IPTG? IPTG is nothing but it is the isopropyl galactoside, and whatever this thing is that this is the inducer of the enzyme actually. And whenever you use this particular thing, if your gene of interest is present within that particular LAG-Z dash, then what will happen? White colonies will be generated. And if your gene of interest is present somewhere else, now we can find out if your gene, this is the gene of interest, it is present in the LAG-Z dash, then we can find out the LAG-Z that is the alpha complementation will not take place, and in the absence of the alpha complementation, you can find that white colonies are generated, and it means that if you get the white colonies, it means that these are the recombinant. It has this particular bacteria has got a recombinant plasmid actually. And in the second occasion, if this is the selectable mark, this is the LAG-Z gene, and here you can find that your gene of interest has been inserted at some other position away from this particular selectable. That's why even if you use this particular technique, if you use the XGAL and IPTG, what will happen? It will produce the blue colony, that is the beta galactoside, that is the protein enzyme will be synthesized, and because of that particular synthesis of the beta galactoside enzyme, you can find that the blue colonies will be formed. And in the third consequence, that is the no insertion, neither it is present in the selectable marker nor it is present outside the selectable marker, then also we'll get the blue colonies. If you get the blue colonies after this particular screening experiment, then it means that there is no recombinant of foreign DNA has not been inserted into the plasmid. And if you get the white colonies, it means that there is a foreign DNA has been inserted. In this way, you can find out this is also another method. Very commonly, these are the two methods which we have discussed are the common methods in majority of the laboratories. And most of the time, you can find out majority of the scientific community, they depend upon this particular like that, because it is much more easier and much more convenient than the antibiotic this thing. That's why this is the way in which the recombinant cells are identified. And with this, we are going to stop here, and thank you very much for your patience.